In this video, we will be discussing about the reticulocyte count and how it is performed in your laboratory. Our learning objective is to identify the reticulocytes on a blood smear, to perform the method of counting, to distinguish the reticulocytes from the mature red blood cells, and to identify conditions and/or diseases related to the abnormal results of the reticulocyte count. What we need is your extraction materials, so your vacutainer sets, glass slides for your blood smear, microscope, and what we're going to use is EDTA tubes as your anticoagulant. You also need your pipette with tips, so we need 50 microliters and one small test tube. The reagent that we will use is an example of your supravital stain, and that is your new methylene blue. For your procedure, just like what we said in our lecture, you have to place equal amounts of your blood sample and your stain. So you place 50 microliters of your new methylene blue and then 50 microliters of your blood in the test tube. You mix properly and allow to stand at room temperature for 3 to 10 minutes. So this allows the reticulocytes adequate time to take up the stain. At the end of the incubation time, we remix the preparation and then transfer 50 microliters of blood stain preparation to a slide and prepare a wedge smear. So allow it to dry and then prepare 2 wedge smears for confirmation of both your calculations. Now after it dries and as soon as the proper area is selected, so what's the proper area or what is the proper objective that we use? We use your OIO. So you count 1,000 red blood cells and the reticulocytes present using the cross-sectional method or the crenellation technique. So the reticulocytes are included in the total 1,000 RBC count. So you have to take note that reticulocytes are counted also as both RBC and reticulocyte. Okay, so you have to count them at the same time. Our formula, use the number of reticulocytes counted over 1,000 RBCs counted multiplied by 100. So our reporting is your unit will be percent. Okay. In this slide, you can see your reticulocytes stained with your new methylene blue. So they are very distinct. You can see them very clearly. They have parang may mga laman sila sa loob, parang granules sa loob. So that's how you identify them. That's how you count them.